Aliens and two lectures on Mendelian genetics. This is kind of fun stuff. I think most of you will kind of enjoy this.
All right.
So first we'll talk a little bit about Gregor Mendel. And then I'll talk about some of the things he did. What he showed, what we know now. And then we'll talk about experiments. And variations. Okay. First of all, questions. Any questions? Any questions? No questions. All I can say is attendance is going down. The lab, the first Monday and Wednesday, I know you guys are in Tuesday, Thursday lab, but lab attendance is really low. I hope you guys are in Tuesday, Thursdays are showing up at lab.
Yeah, we do.
A lot of these things
They run like hell
cemented,
once they can, but they show up.
Presumably cemented in your brain. You know? But anyway, no questions? Yeah, go ahead, please.
I don't know if you can.
Well, go ahead. Yeah, but
there has been only one lab that I missed, and from what I understand, there's no way to remedy that. Is that?
I don't know. You'd have to check with Colette. In terms of treating the lab report again?
Well, yeah, or just making up the points.
Yeah. You have to ask her because I'm not sure what she
She'll
does.
let you turn them in late. It's just hard to do the stuff afterwards.
Right.
You might be able to get with someone that had it, and
As a notes or something.
yeah.
Okay.
I would.
That's what
you
other
do
people
the
have
lab
done.
And then
Right.
ask her if you can turn in late.
That's complicated.
For the lab.
Right. So, but if you can get the results from somebody, so at least you understand what happened. I mean, from our perspective, ours meaning teachers, we consider lab very important because that's where you actually get to do the stuff. You get
hands-on,
and it really is so good for improving your understanding. When I was originally a student, way back 150 years ago, when I was a student, I thought, oh, God, labs, it's a bunch of busy work. No, it's not a bunch of busy work. It's actually very important. So from my perspective, and I can't speak for Colette, but from my perspective, if you can get the results and then understand what actually happened.
Were you ever? What years were you?
It was 150 years ago. About that. It's right before Mendel did his work. Anyway, what I was going to say is
Wasn't an institution yet, but okay.
I post on. You probably can't go on the other site though. I post the stuff I talk about in lab. I post on my CR, but that goes to my students. I don't know if you can get into that. You probably can't. I don't know if it does. I don't know if
She does allow you if you can get with somebody that has done the lab before. She's
allowed you to basically at least review the work, write down the answers.
At least you learn something from
yeah.
that. So that's really what we want. No, I don't think that. No, I don't. I mean, what we the term for not being at the lab and staring at the laboratory is dry labbing. I mean, that's an old term from way back when. And so, we as teachers don't like people dry labbing all their labs, but
Right.
one lab is not a
Yeah.
big deal. That's my theory.
Yeah.
7 labs becomes a big deal,
Yeah.
But
I agree.
but talk to Colette. I think
Okay.
she's pretty understanding. She's a lot nicer than I am.
I don't know about that, but
No, she's a really nice person. I'm a mean person, you know. But anyway, okay. So, okay. Well, let's talk about Gregor Mendel. Actually, interesting person. Actually, Gregor wasn't his original name. His original name was Johann. He was born in what is now the Czech Republic. It wasn't the Czech Republic back then. It was
Czechoslovakia.
part of the Austro-Hungarian Empire and all that. He was actually of German background, and he became a monk. When he became a monk, he took the name Gregor. So he was trained a fair amount in physics. So he had the hard sciences, although he did take some. He did learn about the people that were doing some genetic kinds of experiments. People have been doing genetic experiments for thousands of years. I mean, how do we have domesticated dogs? They're originally from the wild population of wolves, but people,
they domesticated
selecting
us.
for those that are a little bit more gentle and all that. Eventually, they have this breed that likes humans and humans like them. Yeah, go ahead.
If you, I don't know if anybody's been watching Cosmos on Fox.
Oh God,
They domesticated us. It's the other way around.
Yeah, well, that's probably true.
Dogs domesticated
that's
us.
Probably true. They domesticated us. It's probably true.
They got free food by not threatening us.
Yeah, by not being too rough on us.
Yeah.
Come on. But same thing. Breeding of plants and everything. People have been doing these sorts of things for years, but they didn't really understand what was happening. He became a monk. He was in a monastery in a town as I say, now the Czech Republic. I have a feeling he didn't have a lot to do. I can't speak for monks in general whether they have a lot to do. I think he was probably bored. This is my own interpretation. But he started doing various experiments. He wanted to do some breeding experiments, and he had some teachers in college, that had done various breeding experiments. So he wanted to do some. The funny thing is, he started by doing some breeding experiments, sexual reproduction type experiments using, I think, rats or mice or something like that.
Maybe because he wasn't breeding.
Yeah, well, he wasn't breeding because he was a monk. You're right. So he wanted to do these sexual reproduction experiments with rats or mice, and one of the higher up people in the monastery said, "No, you can't do anything on sex." So then he started working on pea plants. But here's what really. I mean, we're going to talk about what he did, and what he did was very important. Early work to improve our understanding of what's happening. But the other thing he did that is really important is he introduced mathematics to biology. And by that I mean he did experiments where he actually did, counted things and then came up with laws dealing with basically mathematical sorts of things. And I think he did that because he had background in the physical sciences. Before then, biology is mainly observational, as I've said many times. People could do experiments, but they never quantified things. Then they started quantifying once he did. Anyway, so his work is good. The interesting thing about his work, we're going to talk about his work. The interesting thing about his work, he published it. It wasn't widely read, and most people that read it thought he was just talking about breeding experiments, but not understanding the genetics aspect of it. And so we didn't get a lot of publicity. That's sort of a weird word to use, but it wasn't probably read a lot. It wasn't really understood. And it was in the 1860s that he did his work. So he did his work. So we'll say work. I can give you. I was going to say in the 1860s if you want an actual date. 1856 to the 63 if you want to be technical. Work 1856 to 1863. So he did his work, middle of the 19th century, early 20th century. Three, I guess, different groups of scientists started doing genetic kinds of experiments. And they were coming up with these interesting ideas about what's happening, and they want to publish. But when you do an experiment you're going to publish, you will always look up and find out what's been published about this before. So we're talking 1909, 1910, 1911. These different, two or three different groups of scientists did this separately, did some really good work. They say, did anything like this published before? And they go back, oh my God, 50 years ago, 60 years ago, someone had published work very similar to this. Oh, no one remembered about it. They didn't know, they didn't have knowledge of it. But sure enough, he definitely beat them to the punch on this one. And so they gave him credit. They said this was discovered, over 50 years ago by Gregor Mendel, although people didn't realize that at the time. For those 50 years, the importance of his work. So Mendel is given the credit for this because he did do it. He did it early on. So when we're talking genetics, we're talking sexual reproduction, and this, whether you're talking plants, animals, doesn't matter what. But you're talking about sperm and egg uniting, forming a new organism. But see, they didn't know about what was happening, and so what they thought. Let's just take animals because it's easier to talk about animals in some ways. And so take animals. They thought that what happened with the offspring was some blending, and they actually thought that in the blood there was some blending of blood and all that, which doesn't happen, of course. But they didn't know, so they thought there was some blending of traits, and that's a term that they often use, like a trait. And so there was a blending, is what they thought. Well, what Mendel showed, let's just write down some of the things he showed. We'll talk about how he showed it and what it means, but let's actually write this down. So Mendel showed 4 things I'm going to put down here. One, that there were units of genetics. He called them factors or something like that. We now call them genes. Okay, we call them genes. That wasn't a term that he used, but we'll use the modern term. There are units of heredity that or genetics that are, they're basically some kind of discrete unit, some kind of separate physical unit. He didn't know what it was. They didn't know anything about DNA. They didn't know anything about chromosomes. He didn't. But anyway, that's what comes out. But so he showed that the units of genetics, and as I said, we now call them genes, so I'll use that term. The units of genetics are material objects. Material elements, whatever, something like that. And they're separate. Separate from each other. In other words, the fact that they are some kind of material thing, there's no blending. You don't get a blending of genes. We'll talk about the expression of genes later, and there are times when you get a blending of the expression of the genes, but you don't get any blending of the genes. They are separate units. They're separate units that can't be transferred from one generation to the next. And that's what I mean by they're separate and they're material, meaning that they are specific elements. Okay. Number 2, he showed that genes come in pairs. Okay. He showed that these genes are maintained through many generations. Now we're going to talk about how he did all these experiments so it all makes sense. Genes are maintained through many generations. And for genes come in pairs, but when you're forming gametes, meiosis, right? We didn't know about meiosis, but when you're forming gametes, the genes, these pairs separate, and only one member of each pair goes into each gamete. So I'm going to do it as a gene. We know it's actually the entire chromosomes that are happening, but the chromosomes are made of genes. We know that. So I'll say gene pairs. We'll do it this way. Gene pairs separate during gamete production. Gene pairs separate during gamete formation. In other words, when you're forming eggs and sperm, each egg is one of each gene. Each sperm gets one of each gene.
He knew this?
The pairs don't go together. Say it again.
He knew this? Or he just...
He showed this. He showed this. Well, and you'll see how he showed it. He's not... he didn't know about how gametes form, but he did the pollination, and we'll talk about that with the bee.
Okay.
So so this is the kind of thing that he was able to show. All right, well, let's talk about just put it in the terms we already know about, of course, what we know now. Of course, we can just do that. What we and you know all this, and we've discussed this. What we know now. Okay, we'll say one. A gene is a stretch of DNA which has the information to tell how to make
one amino acid.
One
protein. Well, see, I got marked wrong.
Codon is for one amino acid.
Ah.
Codon is 3, bases. One amino acid, one codon for that. Okay, so a gene. But see, they didn't know this, and he didn't use the word gene. He talked about, I think he used the word factor, whatever. In German, I think the word factor. He talked about factors being transmitted. They come in pairs and all that. But I'm using the more modern term, genes, because that's what we know the term. So we know that genes, or I'll say a gene. We'll say a gene is a stretch of DNA, or whatever you want to say. That codes, I'll say codes for one protein. Now remember, this is the simplest way to define a gene. You can get more technical, but we don't need to for our purposes. Okay, we know that genes come in pairs because there's one of each pair on the word we used last time, homologous chromosomes. Okay, so we're following what this is. So number 2, genes come in pairs. Okay, we know that a pair of genes, each member of a pair of chromosomes is on each member, or the pair of each member of a pair of chromosomes. God, this is genes here. Well, it's hard to make a decent sentence out of it. I'm having a little bit of trouble. Each member of a pair of genes, what I wanted to say, not chromosomes. Okay. Each member of a pair of genes resides on one of the 2 homologous chromosomes. Okay. As it resides on homologous chromosomes. That makes sense. That makes sense what I'm saying. In other words, we're talking about a specific pair of genes. One will be on mom's number 1 chromosome. The other member of the pair is on dad's number 1 chromosome. That's all I'm saying. Okay. Dad's number 1 and mom's number 1 are homologous. Okay. So, but we know this now. He didn't know this now. He didn't know this. He didn't know about chromosomes. He didn't know about this. Okay. We know that genes are maintained through many generations. We know that is true because DNA is replicated exactly when cells divide. So it'll be true for the generation of, just within an organism, or when you're making gametes and, going to new generations of offspring. And then the last ones, we know that in meiosis, where gene pairs separate during gamete formation, we know that in meiosis, we know this from the last lecture. In meiosis, homologous chromosomes, remember homologous chromosomes are joined together to form tetrads, but those tetrads split, we get that reduction division. So homologous chromosomes in meiosis, homologous chromosomes which we say separate from each other. And this happens in meiosis 1, right?
And also in meiosis 1, they exchange DNA.
We get the crossing over.
Crossing over.
Yeah, exactly. But we'll talk about that later, because he wasn't dealing with that. I mean, he didn't know about that. Okay. So anyway, so as I say, we can understand what he said, more than he knew these things, but without, he didn't have all obviously the biological background that no one had back then. It took 100 years before people understood these things. Well, let me talk a little bit about what he did. He worked with a pea plant, Pisum sativum, just a common pea. And it turns out that this common pea plant self-pollinates. Most of the time, about little bees and, well, about the birds and bees. Yeah, of course you do. But the bees come and they gather pollen as they're getting nectar. They get pollen on their back. They go to another plant, and the pollen can be used to fertilize the other plant. Okay, we know about this. This is common. But some plants, such as the pea plant, so Mendel worked with peas. Pisum sativum is the name of the genus species. Work with peas, and they self-pollinate. This is kind of an important idea.
Meaning they're hermaphroditic organisms.
What happens is that the stamen and the anthers, the parts that do that, they're all within a little what do you call it? A little container kind of a thing. And so, the pollen of the same plant fertilizes itself. I mean, it's rather than having some insect coming in and transferring the pollen from this plant to another, it's just the same plant is pollinating itself.
But it always does that, unlike something that hermaphrodites, which is typically male and female within some.
No,
okay.
I mean, all plants have most all plants, I guess. I don't want to say all of them.
Yeah.
But anyway, they have the male and female parts, But that's not true of all plants. Some are one, some are the other.
Yeah, but they will hermaphrodite.
But a lot of them have both. But anyway,
the
point is that there are no insects involved here that will transfer pollen from this pea plant right here over this pea plant. This pea plant right here self-pollinates. This pea plant over here self-pollinates. Okay. So, pollinate, this is not very well spelled, is it? Self-pollinate. So, and as I said, where all the pollen is formed and everything, that is all, kind of, let's see, it can't be, you don't have insects coming in there because it's all kind of enclosed, which meant that when he decided to do some experiments, he could cut off the parts that have where the pollen is going to form and deliberately take pollen from another plant and introduce it, and not have to worry about some insects coming in and making massive experiments. Yeah. So that turned out to be a very good way of doing it. He can still deal with these things outside where insects are, but they're not going to interrupt. So they self-pollinate. Thus, he, Mendel, could deliberately pollinate them with whatever he wanted. And this is kind of an important idea, just for him, for his work. And he could introduce whatever pollen he wanted, then he could see what kind of results he got. Blah, blah, blah. Okay. Now here's the thing, because they did self-pollinate, because they self-pollinated, they bred true. They always, and I should give you some little terms. I'll give you terms as we go along because, he found out all kinds of things, so we want to introduce some of his words. So we'll do them as we need them. So let's take the parent. Let's take this idea of they self-pollinate. Say, okay. Thus, the plants bred true. Meaning
maintain.
The offspring were always identical to the parents.
Mm-hmm.
Which makes sense if they're self-pollinating. Always going to get the same plant every time. Let me just introduce a word just for we'll use it in the future. Maybe this lecture is not on Monday. They'll talk about the P generation. Now that P, large letter P for parental. So they'll talk about the parental generation, but they'll just label it P. And they'll use F1, F2, etc.
Phenotypes.
And F1 stands for filial, which means
that would be P
offspring,
anyway.
Means, when you talk about filial, you're talking about your children, your offspring. So F1, this would be the first filial generation. F2 would be the second filial generation. So let me show you. Here's the kind of thing we're talking about. This would be the first filial generation. We talk about the P generation or parental generation. F1 would be the first filial generation. F2 would be the second filial generation. And what we mean by the filial is the offspring, okay? So when we're talking about breeding here, we're going to say, okay, if you're crossing at one parent to another parent, we'll use the letters. We'll go P cross P. So that's just crossing of one parent with another parent. Okay, that's all that means. But then what we get out of that, we would get the first filial generation come out of that. We get what it's F1. So from that we get the F1 individuals. That's the first filial generation, first offspring of that parental cross. If you happen to cross an F1 with an F1, we would say, okay, you cross an F1 with an F1, then we get the second filial generation. That's just the way we do it when we're making a little diagram. So that's all we're talking about when I'm saying something like, if I use those letters, that's what we're talking about, just breeding experiments. Because we're going to talk about these, as I said, if not today, we'll do it on Monday, but we'll talk about the breeding experiments we did. So here's the thing, Mendel worked with these self-pollinating plants. But when he looked at these pea plants that were breeding true always, there were various traits that he studied. Okay, so we don't need to make a list of all the traits, but let's just say some of them. So Mendel studied 7 traits. And I'll just give you just a few to show you. Mendel studied for example, he studied seed shape. I'm going to say what they look like in a minute. He studied seed color. He studied pod color. He studied, he studied tall and short. He studied, whatever. He studied 7 different things. I don't even, it doesn't matter.
No distance.
But for example, the seed shapes, seeds could be, some, we'll say, some were smooth and some were wrinkled. But the ones that were smooth, the ones that had smooth seeds, they always had smooth seeds. The ones that had wrinkled seeds always had wrinkled seeds. Okay, see what I'm saying? They read true. Seed color, they could be green or yellow. And the pod color could be green or yellow. So we'll say, so this is seeds. Some seeds were green and some were yellow. Same thing for the pod color. Some were green, some were yellow. So okay, and then there were 4 other traits that he studied. But the point is that whatever trait he was looking at, these, since they were self-pollinating, they were always breeding true. So he said, let me just try an experiment. I mean, I don't know what he said, but I'm hypothesizing. He said, I'm going to try something. Since they're always breeding true, let's see what happens if I take, let's just take one of these, seed shape. Let's take some peas that have smooth seeds and cross them with seeds that have wrinkled, plants that have wrinkled seeds. Okay, so let's do that. See what happens. And he could do this because, as I said, since they're self-pollinating, he could remove the pollen from both of those plants that he's dealing with, and saying, okay, now for the one plant that had smooth seeds, I'm going to introduce some wrinkled pollen. So I'm calling it wrinkled pollen. Pollen was wrinkled, but you know what I mean. And for the wrinkled plants, he introduced the smooth pollen. When he did this, so let's do this. So we'll say, so for the parental generation, he crossed a smooth seed. One of the plants that had smooth seed, he crossed it. He deliberately introduced pollen to one that had wrinkled seeds. And the F1 generation, all the offspring had smooth seeds. So now probably a lot of people who might have tried this experiment would have said, well, okay, so we don't know what causes wrinkled seeds, but they're gone now, Well, they're not really gone because remember what I said? He learned this by doing experiments. He said, let's try another experiment. Let's actually take these F1s. Let's take these F1s and cross them with each other. So let's take these. Right, one of these F1s with F1s. Okay. When he did this, and remember, now he did hundreds of experiments, so he had hundreds and hundreds and hundreds of plants that he could study, and then he would count. In this case, he'd count we're looking at what seed shape. He'd look at the seeds of these and say, okay, because what he found out was some of the offspring of the F1 crossed with F1, some of them had smooth seeds, some had wrinkled seeds. So the wrinkled seeds reappeared in some of the plants. And roughly it came out to be about, there were 3 smooth seeds per 1 wrinkled seed. Now what I'm saying here is there's 3-to-1 ratio of smooth seeds to wrinkled seeds. And now we're talking hundreds of offspring. Okay, he took a whole bunch of these F1s and did the deliberate pollination. One F1 with another F1, another one, and then counted all the seeds. And so the numbers are, he had hundreds and hundreds, maybe 900 of smooth seeds, and whatever, say 300 or something wrinkle seeds. Whatever. I mean, they weren't exact numbers. They were like, it would be like 937 and 644. I mean, whatever. But he did the experiment over and over and over again. But what happened is that, first of all, they didn't, when you did the smooth seeds and you cross those with wrinkle seeds, all the offspring were wrinkled. Smooth, sorry, wrinkled smooth. But when you did cross the F1 with the F2, F1 with F1, sorry, then the F1 cross F1, the F2, in this case, we got about 3 smooth seeds per 1 wrinkle.
So 75%.
Which means that the wrinkle didn't disappear. Whatever that trait, that wrinkled trait, didn't just disappear. It was, quote, hidden. I mean, we know what it was, just wasn't showing. It wasn't being expressed, but we'll understand that in a minute.
Is that what a recessive gene is?
Well, that's what he started using. I think he used those terms. Those are the terms we use now, and I think he started to use those terms. I'm not sure exactly which specific terms he did use, but we now use those terms. But let me give you some terms now, and then we will say exactly this. So first of all, we're going to use, these are modern terms, and these are terms you need to know. We'll talk about the phenotype. So we will be introducing these as we go along. Phenotype versus genotype. So phenotype, well, let me do the word genotype first. That's actually a little easier. The genotype says what genes you actually possess, what genes that organism has. The actual genes that are in the organism. So the genotype. And right now we're only talking about, we will be talking about the genes for smooth seed, the genes for wrinkled seed. That's what we're talking about. But so genotype just says, hey, what genes do you have? Phenotype, the word phenotype means how are those genes expressed? How the genes are expressed. So whatever the genes they are, if you're a smooth seed, it's being expressed as smooth. Whatever the genes are that you have, if you have wrinkled seeds, the expression of those genes is wrinkled seed. Okay. But then when he did these experiments, he goes, wait a minute, this is weird, I took smooth seeded plants that always breed smooth and always give you smooth seeds, and I crossed them with wrinkled seeded plants that always give me wrinkled seeds no matter what. I crossed those, and all the offspring were smooth. And then he says, but let me just try crossing some of these with each other, and sure enough, most of them are smooth seeds, but some of them have wrinkled seeds. So, this is unusual. So then we come up with these ideas of dominant versus recessive. Dominant genes versus recessive genes. Okay. So a dominant gene is expressed if you only need to have one copy of it. Now remember, we're talking about genes coming in pairs. Remember that genes come in pairs. So the genes for the seed shape, you inherit 2. Well, if you're a pea plant anyway, you inherit 2, one from the mom, one from the dad. But in terms of the dominant gene, it turns out you just need to have one copy of whatever that gene is, and you will express whatever the dominant trait is. So the dominant gene is expressed with 1 or 2 copies of the gene. Now notice 1 or 2. Recessive gene, that's only going to be expressed only if there are 2 copies. So let's look at what we did now. Let's see how this worked. And this is the kind of thing I want you to kind of understand in terms of how this all works out. So let's do this. So And we want to use these terms, dominant and recessive, because this turns out to be very important. So the way people do it, I don't know how. I'm not sure how, if exactly how Mendel did it, but I'll show you the way they tend to do it nowadays. It turns out that of these two traits, smooth is dominant, smooth seed is dominant, wrinkled seed is recessive. So what they do, well, let's just actually write that down. So it turns out that with these pea plants, smooth seeds is recessive. It's dominant, sorry. Wrinkled seeds is recessive. Now here's the way we tend to write this. This is the way it's usually done. This is what I want you to learn. So they'll use a letter for whatever the trait is. Okay. Now sometimes it gets difficult because a big S and a little s might look alike, so you have to be kind of careful. But they tend to use a big S for the dominant trait and a little s for the recessive trait. And you go little. Why don't you use it one S and a W? No, that's not the way they do it. We'll say an F1 and F1. No, let's not use F1. So okay. So the three possibilities of what the plant might have for seed shape. We'll say so. Three possibilities for the genotype of the genotype for seed shape. Now here, and this is the thing you just have to understand. This is the way they do it. This is just the common way of doing it. You could have big S, big S, big S. I'm going to make a little s like a little written S so you can see it. Or you can have little s, okay? But they use S. So, and the big S, and the big S, we'll say the big S is for the smooth, and the little s is going to be for wrinkle. In terms of the phenotype, right? Well, let's just, I don't remember what the phenotype. But the big S is the dominant trait. The little s is the wrinkled seed. That's the recessive trait. So three possibilities for the genotype for seed shape. Now, what did we say? The dominant genes are expressed with whether you have one or two of them. And what's the dominant one? The dominant, we said, is smooth. So so some of those individuals might have two of the big S genes. Some might have one big S and one little s, but they'll both express the dominant trait, because the dominant trait will be expressed if you only have one of them. So both of these, this is going to be, I'll write underneath here, this is going to be the genotype. Okay, this is the genotype, and over here we'll put the phenotype. So what I'm saying is there are two possible genotypes that should give us the dominant trait. What did we say the dominant trait was? Smooth. So both of these will give you smooth seeds. In other words, if a plant has one dominant gene, has two dominant genes for smooth, they'll be smooth. They have one dominant gene for smooth, but one of those recessive genes for wrinkled, they'll still come out to have smooth seeds.
How do you know which one's recessive and which one's dominant?
Well, only by doing experiments.
And if the dominant one
shows up more.
Yeah, the one that will show up like that. I mean, you're right. You can't tell just by looking, talking about it. You have to have done experiments to see which one is expressed. It's a good question. But the point here is that if the genotype is two big S's or big S little s, You have to be careful because people want to talk about it. Well, it could be SS or SS. Oh, no, you can't say SS. Big S, big S, big S, little s. Say something like that. So you can see the difference. You'll still get a smooth seed. And whereas if you have two little s's, those plants will have wrinkled seeds. Of course, you can imagine. What if you went back to that parental? If you went back to the parental generation, they were always breeding true. So the parental generation that had smooth seeds, they all had big S, didn't they? And the parental generation that was breeding, always giving you wrinkled seeds, they had little s, little s. But what happened in the F1 generation when he crossed the smooth with the wrinkled? So let's actually look at that and see what would happen. And now here's where you just are showing the possibilities here. There's a way to do this with respect to what's called a Punnett square. We'll do a Punnett square in a minute. Let's just do it on ourselves just without a Punnett square. Same thing, but so if this guy always bred true, the smooth seed, then that would have been equivalent to having a big S, right? And what about the wrinkled one? He would always have a
small s.
Breeding true, he'd have a small s and a small s. So here are their genotypes, right? That would be the original parents because they were always breeding true. And so their phenotypes, smooth, wrinkled. Their genotypes, the smooth one had two big s's, the wrinkled one had two little s's. Now what about when you make gametes? Those are the parents. You make gametes. With a gamete, you're going to have one of these two s's in there, or one of these two s's. So all these individuals, all their gametes had a big S. That's right, one big S. And the wrinkled ones, all their gametes had one little s. So if he crossed those, what are all the F1s going to have? They're all going to have a big S, a little s. And what is going to be that's the genotype to all the F1s. This is the parental, remember, and this is the F1 now. All the F1s have a big S and a little s. So what's their phenotype? Smooth. Yeah. They're all going to look smooth. Even though they do have that, what we now call a recessive gene. This is the dominant. Big S is the dominant gene. The little s is the recessive gene. But what's going to be expressed is the dominant trait, which is smooth seeds. Okay, so that makes sense then what happened in the F1 generation. We'll come back to that idea. Roughly, I'll give you the rough idea right now, but we will come back because it's going to be more complicated than I'm going to say. But
prevalence versus mutation.
The simple explanation is a dominant gene says to make something. And so it may say, hey, make a particular enzyme that somehow causes the seed to be smooth. Whereas the wrinkled one says, hey, don't make it. Something maybe the mutation says don't make it. Yeah. It's not making it, then it came out all wrinkly. So in simplest terms, but not complete, is not the total answer. In simple terms, the dominant gene says do something, recessive gene says don't do something. So if in this case, if that's a simple explanation, this says, hey, make it smooth. This says don't just hang out, don't do anything. Well, it's not going to have any effect. If it's saying don't do anything, it's not having an effect. Because this one is saying do it. And what you're doing is making smooth seeds. Is that easy enough to get that? It's a good question, very good question, and it's something I want to come back to because it's a little more complicated. All right, so this explains, but of course, Mendel didn't know this when he first did it. He wasn't sure about it. But now he said, okay, he was able to kind of figure this out later after he'd done more experiments. He said, let's take all these F1s, a bunch of them, and cross them with each other. So we take these, so what we're crossing in the F1 now is we're going to do an F1 cross F1, we're crossing something with a big S and a little s with another big S and a little s. And now we're going to get the F2s. But again, we've got to look at the gametes. What kinds of gametes is this guy over here going to make? Half of them will have a big S, half will have a little s. Over here, half of these guys will have a big S, half will have a little s. See what I'm saying? So let's make a little Punnett square now. they call it a Punnett square after the guy who first started using these. He was an American geneticist. I think he was American. Oh gosh, I don't know, maybe 1920s or something like that. I forget when he lived. But anyway, he's the one that said, hey, this is an easy way to do these is to make a little square. And so here's what he would do. I'll cross. I'll just erase this. You guys know about the exam. So he said, what we'll do, make a little square. In this case, it'll be a 2 by 2. And we'll do this. We'll do this called a Punnett square. If I remember correctly, it's 2 N's and 2 T's. That was his name, as I said. So in this case, it's going to be a 2 by 2. I wish I didn't do it big enough. I'll do it big like this. Okay. And what you do is you put what you put in these outer things here are the what gametes can be made. It doesn't matter whether you put the male on the top, the female on the side, or the female on the top, the male on the side. It doesn't matter. But so here you're going to have what genes, and I'll say what genes are in the gametes. What genes are in the gametes? And you're going to do it across this way and down this way. And so we're crossing what? A big S. So we're crossing a big S and a little s. So I'll just do this. So we can have this one coming up here, going like that. And this one we could have coming down, okay? So what are the possible genes in the gametes? How many different kinds of gametes can this guy make? Different gametes in terms of this trait. Exactly 2, right?
2.
Half of them will have a big S. Half of them will have a big S, and half will have a little s. I wouldn't use, if you're going to do s's, don't do s and s, because you'll start, as you start writing fast, you'll forget which one was big and which was little, because I'll start to like this size, and that's why I'm making the 2 s's look different. You have to be really careful sometimes with the letters. So half of the gametes will have a big S, half will have a little s. What about this individual here? Exact same thing, right? Half will have a big S, and half will have a little s. So those are on the outer things. They're not on the inner things. Those are on the outer. These are the possible gametes going across from one individual to the possible gametes of the other individual.
Oh, yeah.
See what I'm saying? Does that make sense what I'm saying?
So that's
now
how
you just look at this and you go,
75%.
hey, if you had to get a cross between this gamete and this gamete, what does this guy end up with?
Smooth.
Big S. Well, let's just do the gametes. Big S, big S. What if this guy happened to mate with this one?
Yes.
You get big S, little s. What if this guy happened to mate with this guy?
Little s,
Big S, little s. These two mated?
Little s,
Little s, little s. By the way, they always put the big letter first, the little letter second.
Ah, okay.
It's not an absolute, but that's just the way it's always done. So, if you're doing a whole bunch of, if you only had one offspring, you could be any of those. But if you're doing hundreds of them, then you expect it to come out in this ratio. And what are these? These are the genotypes that you would get. What's the phenotype of this?
Smooth.
Smooth.
So 75% of the time, the dominant
And
gene.
these two are also smooth, right, in terms of the phenotype?
Yeah.
Smooth. And these, of course, are wrinkled. Because you have to have both of the recessive genes to be wrinkled. And so if you did a whole bunch of experiments, you'd come out 3, 2, 1, smooth, 2, wrinkled. That make sense?
Yeah, but does that mean for all genetics, 75% of the time the dominant gene wins out?
Okay. In general, that would be yes. You would say that, but there are. This is just the simplest interpretation.
I get that life doesn't.
But yeah, that would be the basic idea.
Okay.
If you crossed, if you are crossing. Let's see.
The first two phenotypes, or the first two.
The dominant. When they call this the homozygous dominant with the homozygous recessive. We haven't used those words yet. But
Okay.
Said them. Then you get what they call a heterozygote. Heterozygotes. All the F1s are heterozygotes, one of each. So if you cross these guys like this, then you should come out with the 3-to-1
Okay.
ratio.
Okay.
Yeah. Actually, I'll use those words heterozygous and homozygous. Yeah, go ahead.
Well, for the F1, when he crossed the parent and the wrinkle seed.
When I originally crossed the two parentals.
I know, but like at first you cross the P times the P and
right.
you get the F1.
And that gave me all of them were this, all the offspring.
And all of them were smooth. How did he? Oh, because
he just took a. Yeah, he was doing hundreds of these things.
I know, but if he was doing hundreds, wouldn't some of the parents be a big F and a little F?
But no, but he wasn't. But I mean, he's not looking at these now. He's just saying, hey, let me just take all these plants. He's forgetting these guys now. He says, let me just take all these and offspring, and let me just cross one offspring to another.
I know, I just don't get how all the F1 became smooth seeds. Because how could he ensure that all the parents were like all? Yeah.
All parents were two double S.
Yeah.
Big S.
Yeah, instead of a big S.
Because these have been breeding true for hundreds of years.
Self-pollinating.
So
when he crossed him, first time the two genes been crossed.
And he probably kept watching them, but they were breeding true, for years. So, and he didn't know for sure about their genetics, because he hadn't done any experiments yet. But it turns out because they had been breeding true, and they're self-pollinating. That was the trick. They're self-pollinating. That turned out they had to be this and this, So then all the offspring had to be this. Once he did the experiments, I mean, he didn't know this. He just did the experiments, and they all came out to be smooth. And then he said, well, now let's do another thing. Let's take all of these guys and breed them. And he goes, ooh, some of them came out to be wrinkled. There's no wrinkled in these. And yet some of their offspring came out to be wrinkled. So that's when he said, gee, there must be two separate elements. We call them genes now. And, they're individual kind of elements. There's no blending going on, and they're particulate or separate or whatever you want to say. And they're not, they weren't lost in this F1 generation. They just weren't being expressed. And then, if you got two of those little S's, they will be expressed. So, I mean, this is all new, remember. This is all very new. He didn't know these things. He didn't know what to expect. Maybe he was just playing around and go, hey, this is weird. Now then he said, this is exciting. Let me try some more. Yeah, go ahead.
So prior to him crossing them, they were always both big S and both little S. Yeah. So there never could have been any
because
diversity.
They were breeding true, because they were self-pollinating.
Yeah. Okay. That's a neat trick. Or a happy accident, whatever.
Yeah. Now, interestingly, he studied 7 traits. The 7, the piece of satiatum, that P, has 7 pairs of chromosomes. And each trait he studied was on a separate chromosome. He didn't know this. And what they think may have happened is he may have tried some other traits, and he was getting some weird results. He probably goes, I can't make sense out of these weird results. Let me just stick with these 7 that are making sense. Mm-hmm. And then that's good. I mean, that's good because, you don't want these complicating things. You need to build up. If you start with a complicated thing, you go, can't make sense of it. So it turns out that it was probably very fortunate that he picked these 7, or probably he tried this so the others and didn't work out. But anyway, that's it's sort of a weird thing. 7 pairs of chromosomes. All right, so I gave you these terms, homozygous and heterozygous. Let me just give you those. So again, I want you to know these terms because these are so homozygous. Now we're talking about homozygous for a given trait, or homozygous for
meaning
a
same
given gene.
Same gene.
And homo means the same. This means they have the organism has 2 of the same gene. It doesn't tell you which one. It could be 2 dominant ones, could be 2 recessive. So the way we distinguish, we say, hey, there's homozygous dominant or homozygous recessive. Heterozygous
means different.
Just means you have 1 of each, or two different genes. I'll say 2 different genes. So, for example, so each of these. So this one right here, the 2 big S's, that would be homozygous dominant. And the two little s's would be homozygous recessive. Okay. And then, of course, the S1, the F1 was all big S, little s. These are both heterozygous. I mean, all of them were heterozygous.
Because they have different, genes.
Heterozygous. Okay. So again, these are words that are just convenient to use, so that when you're talking about it, you don't have to. Oh, it has the two big ones. You can just use these words and make sense out of it. Okay. Does this make sense? What's happening so far? I mean, this is pretty weird. Now, he didn't come up with this immediately. I mean, he did a lot of breeding experiments, a lot. And I don't know how many months and months and months it took him to do it, but because he would then, these are the seeds. He'd have to open up the pod and look at all the seeds in there, and then he'd count, from all the offspring. And he'd count. So he had hundreds and hundreds and hundreds of these experiments where he's counted so many. And then once he got the numbers, he's going, oh, do these numbers make sense? Is there anything? And then he said, well, gee, he never would get exactly 3 to 1. That
Those
would
are close.
be very unlikely to get exactly 3 to 1, but he'd get like 2.96 to 1 in one experiment. Another experiment he might get 3.23 to 1 or something like that. But the point is,
the mean came down to
approaching 3 to 1, but by being almost an integer, and you figure by just normal statistics, you're not going to get exactly 3 to exactly 1. Then you're going to, he was able to come up with these ideas about having these things come in pairs and having them separate individually when you're making, he didn't know the word meiosis, they didn't use that term. We know it now.
What would the F2s be? Can it just be an
So the F2, these are the F2s. These are the F2s up here. Here, sorry. Well, I have it here. This is the F2. Okay, this is the F2s right here. But here I'm illustrating it here. Does that make sense what I'm saying?
So it could be either one of those?
Well, yeah, so the F2, you got 3 smooths to 1 wrinkle. But if you said, well, what about their genes or genotype? Oh, well, a fourth of them are homozygous dominant, a fourth of them are homozygous recessive, and half of them are heterozygous. But the phenotype, 3 of them are smooth in 1 wrinkle. So it depends what you're talking about, the genes, the genotype or the phenotype. Does that make sense?
Yep.
Yeah.
It's kind of wild. It's kind of weird the way he did all this. This is great. Now, we're going to get more complicated because this is, a seed shape. But he did the same kind of thing for seed color. And some were green and some were yellow, and one of those is dominant. The green, I think, is dominant. The yellow is recessive. And the pod, had this. I think it's the opposite. I think yellow may be dominant on that one. I don't remember all. Well, the seed color, yellow is dominant over green. Pod color, green dominant over yellow. But whatever, that's not important right at the moment. The important thing is he first did a bunch of these experiments with 7 different traits and found out which is the dominant, which is the recessive. And then he says, what if I do across, and I'm looking at 2 different traits. I'm looking at seed color and seed shape, for example. And, he'd get the ones that are breeding true, and then he'd get the 2 different, the dominant, the homozygous dominance and the homozygous recessive, then he'd breed those. And, it comes out to be 9 to 3 to 3 to 1, which we'll talk about next lecture. But the point is that it gets more complicated, but it still made sense mathematically because it's making sense scientifically. But we understand why it's making sense better than Mendel, but he did a pretty good job, But this totally revolutionized genetics because, as I said before, they thought, oh, things were just some kind of a blending. But no, it's not a blending. They're separate elements, these genes, and depending upon what combinations you have, they'll be expressed in a certain, what we now call a phenotype. What was I going to say? I forget the term. I forget exactly what I was going to say about that. But the point is that the wrinkled gene, it can stay. If you keep breeding these dihybrids here, not the dihybrids, sorry, the heterozygotes, keep breeding those, you'll every so often show the wrinkles. But I mean, that even if you only choose smooth seeds in your offspring, you're still going to have some with that recessive gene. It's not showing up unless it's in, unless you have two of them that are homozygous to show up. So that's what he was saying, is that you don't just lose these things necessarily. They may not be expressed, but the gene will be there. Isn't anyone excited about this?
Well,
I'm teasing you, but this is kind of weird. No,
that's not how I express excitement over science.
I think what I want, I'll give you the first law. The first law, and then I won't go any further today.
Okay.
Because that would be, and then we talk about the second law. So I told you this is 2 lectures. So we did part of his experiment. We haven't done any of the so-called dihybrids, where we're looking at 2 traits at the same time, like seed color and seed shape. We'll do that on Monday. But we can at least talk about his first law. So let's talk about the law of independent segregation. And this we already know is true from what we talked about in meiosis. Well, I'm going to leave this stuff on the board right here. So let's just talk about it here. So the first law, Mendel's first law. And by the way, what we will see next time after we do the second law of the dihybrid crosses and all that, there are examples of genetics that don't follow Mendel's law because of, well, we'll see why. But say blood type, for example, we'll see that there's a codominance factor there, recessive, some genes, and 2 of them are so-called codominant and one's recessive. That way you can get type A, type B, type A, B, type O. We'll talk about that next time. But let's just do independent segregation. And this is something you already know. That in gamete formation, well, before I write that, maybe I'll do this. I know you started writing, and then I know that I changed his mind again. What do you want to say?
So yeah, no, I only have to write later. Yeah.
Wait a few seconds, hesitate till I decide if I like that. Okay, so when we're talking about 2 genes, we're talking about the gene for, say, seed shape. We have 2 genes. They could be big S or it could be little s, but you have 2 of them. Okay, so we'll say of the 2 genes for a trait, we'll say, for example, S, big S or little s for C shape. And each organism, I haven't done that independent segregation yet, I'm building up to it. Of the 2 genes for a trait, for example, big S or little s for seed shape, each organism contains both 2 genes. So of the 2 genes for a trait, when gametes are formed, these 2 genes go into different cells, different gametes. Okay. In other words, each gamete gets only one of those 2 genes, not both. So of the 2 genes for a trait, each gamete will get only one of those genes. We know this happens because, these are on homologous chromosomes, and in meiosis, homologous chromosomes separate, right? So does that make sense when I said this? My own wording here. You can get different wording in the book. The same thing though.
No.
But the whole point is that when you form gametes, each gamete gets one of each chromosome. So it's going to get one of each gene because these two genes are on homologous chromosomes. One's on your dad's chromosome, one's on your mom's chromosome. They'll be separated. That's all independent segregation means. The genes will separate from each other when you're forming gametes. That's all it means.
And gametes is just through meiosis, it's the division of cells, and okay.
Okay, does that make sense?
Yep.
Okay, so what we'll do now. I don't want to start it right now because I might even let you out early. Maybe let you guys out early.
Whoa.
Maybe I will. But anyway.
Don't do it. It's a trick.
but I don't want to start it now because I want to do that whole thing. But we'll talk about the dihybrid cross. We'll talk about taking things that have smooth seeds and yellow seeds and cross them with wrinkled seeds and green seeds and see what we get. Then we have two different sets of genes. And then we'll see how that plays out. We'll use the Punnett square and see how that plays out. And then we'll come up with the second law, which is the law of independent assortment. We'll do that on Monday. Oh my God, I'm going to let you out early. Whoopee. All right. What an unusual phenomenon. Oh, get a study guide. If you didn't get a study guide, get a study guide.
See you guys. Oh, turn off, Dan.